We have now designed our primers. An important method of reducing large quantities of a particular DNA sequence is the polymerase chain reaction, or PCR. So what is PCR and what is its purpose? Genomic DNA in cells has thousands of genes, making it hard to isolate one. PCR lets you copy a specific DNA sequence, like a gene, many times using a simple enzyme reaction. PCR is very selective and sensitive and is able to amplify DNA from just one copy to over 200 million copies. This produces enough DNA in one reaction for detailed study or further work with the gene. What are the components of the reaction? The template DNA. The template DNA contains the target sequence and can be a complex mix. Even tiny amounts of the target DNA in a sample can be used. Oligonucleotide primers. To perform PCR, you need to know the DNA sequence at the target's borders. Short DNA primers matching these regions are made. These primers attach to the single DNA strands and help DNA polymerase copy the target sequence. DNA polymerase. Thermostable DNA polymerases, like Taq, copy DNA and PCR. They withstand high temperatures, up to 1000 degrees Celsius, to synthesize new DNA strands. Deoxynucleotide triphosphates, or DNTPs. These molecules correspond to the 4 nucleotides present in DNA and are substrates for the DNA polymerase. The PCR cycle. PCR uses key features of DNA replication. DNA undergoes denaturation at over 90 degrees Celsius to separate the strands. Annealing of primers occurs at 5 degrees Celsius below their melting temperature for 30 seconds. Polymerization happens at 72 degrees Celsius for Taq polymerase, with 1 minute per kilobase of target DNA. The polymerase must be thermostable to withstand high temperatures. Repeating this process 25 to 30 cycles amplifies the target DNA over 1 million times, producing many copies of the desired gene. Because each step is dependent on temperature, the steps can be executed by simply changing the temperature of the reaction. Fast and accurate temperature shifts are achieved using an instrument called a thermal cycler. You'll be provided template DNA to amplify the AMI-E gene via PCR. Understanding each PCR's components' roles is important. Primers from the previous task, DNA polymerase, DNTPs, buffer, and water. Knowing their functions is key to successful amplification. For the PCR, a master mix will be provided containing the primers, DNTPs, magnesium chloride, Taq DNA polymerase, and its action buffer. You will also be provided with sterile water. Set up one polymerase chain reaction as follows. Taq buffer, 10x. The reaction buffer is 10x concentrated. The concentration of constituents following a 1 in 10 dilution is 10 millimolar tris HCl, pH 9.0, 50 millimolar potassium chloride, and 0.1% Triton X100. Place the reactions in the thermal cycler and set the program as follows. Remove the samples on completion of the cycles. The products can be stored at -20 degrees Celsius. The learning objective is now that you should be able to understand the theory, especially PCR components, and perform a PCR. PCR product purification. GE Healthcare, QIAGEN, and other companies produce mini columns or membranes that facilitate the rapid purification of DNA fragments. The binding material is a silica mix that allows flow-through of other components in the reaction mix, such as small oligonucleotides or primers and buffer components. The DNA binding is pH dependent, and after washing, the release of the DNA from the membrane is affected by changing the pH. For the procedure, add 500 microliters of capture buffer type 3 to 100 microliters of PCR reaction. To obtain 100 microliters of PCR sample, combine the two 50 microliter PCR reactions. Mix thoroughly by inversion. The color of the mixture should be yellow or pale orange. Place a column in a collection tube and add the solution to the column. Centrifuge for 60 seconds at full speed in a microfuge. Discard the flow through from the collection tube. Place the column back in the collection tube and add 500 microliters of wash buffer type 1 to the wash column. Centrifuge for 60 seconds at full speed and discard the flow through from the collection tube. Replace the column in the collection tube and centrifuge for 60 seconds to remove any residual buffer. Place the column in a new 1.5mm microfuge tube. Add 40 microliters of elution buffer type 4 to the center of the membrane. Let it stand for at least 60 seconds and then centrifuge for 60 seconds. The eluted DNA can now be used for digestion. The learning objective from this exercise is that now you will be able to purify your DNA samples away from contaminated proteins and unwanted buffers, ready for later restriction digestion. Now that you've completed your PCR reaction and purified your product, you're ready to move on to the insertion of your target DNA into a plasmid vector.